Another live parasitology session. Today we'll be talking about Toxoplasma gondii. If you haven't watched our recent sessions on parasitology, the different parasites, be sure to check them out. I hope you'll find all of them beneficial. So let's get started with today's session. As I said earlier, that the parasite we're discussing today is Toxoplasma gondii. It is responsible for causing the disease toxoplasmosis. This disease ends with the word osis, which is the characteristic finding of any parasitic disease. Like any parasitic disease will end with osis or asis. So whenever you find a disease ending with these words, you can figure out that this is a parasitic disease. Toxoplasma gondii is a protozoan parasite. It's a tissue parasite. Protozoan are those parasites that are uninucleate, that are single-celled. It is comma-shaped. It is an epicomplexan parasite. Epicomplexans are the parasites that are comma-shaped, that are narrow at anterior end and wider at the posterior end, and they've got different organelles in them like mitochondria, Golgi apparatus, ER, the endoplasmic reticulum, rhabdoid, and others. This parasite has got 3 stages in its life cycle. All of these stages are important. Let me tell you what are these 3 stages. First one is sporozoites, then we've got bradyzoites, and lastly, the tachyzoites. Tachyzoites are also referred as trophozoites. And there's another form that is cyst, which is responsible in the transmission of this parasite from one host to the other. This parasite is also called the cat parasite because it is found in the cat feces. Let's talk about its transmission. The route of transmission is oral fecal route. It is transmitted by ingesting the cysts in raw meat or in food that is contaminated with cat feces. It is also transmitted by transplacental route, like the passage of trophozoite transplacentally from mother to fetus. Infection of fetus occurs only when mother is infected during pregnancy and when she is infected for the first time, which means she has no protective antibody. Cat is playing a major role in causing this infection. Habitat. Let's talk about the different hosts of this parasite. Humans are its hosts. You might be wondering, I've written the word IH after humans. This is because humans are the intermediate hosts. And then I've written the word DH after cats. This is because cats are the definitive hosts. Normally, Toxoplasma is found in cats, but it uses humans as its intermediate hosts and causes toxoplasmosis in humans as well. Now let's talk about the life cycle of Toxoplasma gondii. As we all know, it has got 3 stages in its life cycle: sporozoites, trophozoites, the tachyzoites, and bradyzoites. And there's another stage that is cyst. So let's start with the cyst. Cysts in cat feces or in meat are ingested by humans, and they differentiate into different forms in gut. And then they are able to invade the gut wall. We are talking about the human gut, right? The trophozoites, cysts, they infect macrophages and form trophozoites, the tachyzoites actually. These tachyzoites multiply rapidly, kill cells, and infect other cells. Cysts containing bradyzoites are formed later. Cat ingests cysts in raw meat and bradyzoids exist. Then they multiply and form male and female gametocytes. Now what happens? These gametocytes fuse together and they form oocysts in the cat gut. Now this is the stage when the human cycle ends and the cat cycle begins. And these oocysts are excreted in cat feces. And then human ingests the food that is contaminated with cat feces and gets those cysts again. And then this infection is caused in humans again. And the infection is called toxoplasmosis. Now let's look at the pathogenesis. The trophozoites, the tachyzoids, are actually trophozoites. They infect many organs in human body, especially brain, eyes, and liver. Cysts persist in tissue. They enlarge and cause symptoms in tissues. Severe disease occurs in patients with deficient cell-mediated immunity, like people who are immunocompromised. For example, AIDS patient, the acquired immunodeficiency syndrome patient. This disease is caused by HIV, the human immunodeficiency virus. Now let's look at the lab diagnosis. We now know about the parasite, we now know how it is responsible for causing the disease, but how do we diagnose it? We'll definitely need certain samples. We'll go for blood, amniotic fluid, because this parasite also causes congenital toxoplasmosis in fetus. And we'll also go for CSF, the cerebrospinal fluid sample. Then we'll take these samples and we'll visualize them under the microscope. And we'll see the cysts and the trophozoites under the microscope. Cysts are circular, while trophozoites are comma-shaped, narrow at the anterior end and wider at the posterior end. We'll also go for culturing the parasite.